It's great to be here. Thanks for joining me in this session on searching the literature for new approach methodologies or NAMs. I'm just going to make sure that everything is set here. Okay, great. I'm a basic science informationist at the Welch Medical Library. I regularly help with evidence syntheses, so that's scoping reviews and systematic reviews. I also help folks navigate biological data. So I'm all about searching all the time, have a lot of expertise there. And so that's just a little bit of my background. So let's dive in now to my talk. I'll be partly or a lot of the time we'll be working through my slides, but we're also going to be going live to PubMed and the MeSH database. And what I would love is for you to be able to build a search at the end of this session, a draft search that incorporates a number of key processes for developing comprehensive searches of the literature. So that you'll have a document that demonstrates some skills in developing these kinds of advanced searches, and that'll be a reference point for you going forward to build similar kinds of searches focused on your particular research interests. So let's start a little bit by unpacking the title of this talk. So we're talking about mastering, and so just breaking that down a little bit. Master is to overcome, to master one's fears, and I think this is maybe an appropriate way to frame this, in that I think there may be some anxieties around searching the literature. How does one do it most effectively? Have I done a sufficiently rigorous search? Am I finding everything that I'm looking for? So there's a fair amount of, I think, I won't say fear necessarily, but just some anxieties around those kinds of questions. Have I, done enough, and am I doing it properly? I think, mastery is also about becoming skilled or proficient in the use of something. And an example might be to master a foreign language. And I think this is particularly apt because searching, I think, is, in a sense, its own sort of language with its own kinds of rules and systems. And so we're going to be talking about those rules and systems today of sort of advanced searching. And, mastery is also related to gaining a thorough understanding of something. And this is my hope, that at the end of the class today, you will have mastered 5 key processes for building a high quality search in PubMed that focuses on NAMs. I would say the skills we're learning today are actually quite transferable. So definitely it's going to be focused on NAMs. And I think there are unique things about NAMs and searching for NAMs in PubMed and other databases that we're going to talk about. But there is a lot of transferability of what we'll talk about today to other contexts. Now, to talk about NAMs a little bit. And, what I think is really helpful when thinking about a topic is, and particularly a topic like NAMs, which has many different methods attached to it, many different approaches for these kinds of new methodologies, is to try to think about it globally from a bird's eye view. And certainly PubMed and the MeSH database, which we'll talk about later, helps you do that and is key to searching. But there are other examples out there, including this NAMO ontological schema that I found, which I think is a nice representation of NAMs. So it is this hierarchically arranged set of ideas and components of NAMs. There is the NAM model as kind of the parent term, as it were, and then there are child terms related to cellular systems, microphysiological systems, and in silico models. And then within these child terms, there are grandchild terms. So there's 2D cell culture and 3D cell culture and co-culture, which are sort of living underneath the umbrella term of cellular systems. And within those, there are even another layer of approaches, cell line models and organoids. And organoids in blue, because that's what we're going to focus on today. NAMs are quite extensive. The number of approaches is quite extensive, and the number of terms that would need to be used to capture all of those approaches is quite extensive. And so we're going to focus, in the interest of time, on just one sort of approach within NAMs, which is organoids. Under microphysiological systems, in this schema, there are organ on chip and tissue on chip categories. And then in the in silico model grouping, there are quasar models, PBPK models, digital twin, machine learning models, and metabolic models. This is the new approach methodology ontology and schema, or NAMO, which I found on the bioportal site. And I think it's just helpful as a way to kind of conceptualize what NAMs are. And this kind of conceptualization, this kind of visualizing of the landscape is really useful in searching, because this gives you the raw material, as it were, for the search, the raw material being the terminology. So you need to have terms in order to search. And this kind of representation is a way to visualize those terms and to think about the relationship amongst terms as a way to structure your search and to get terminology for your search. And we'll be looking at another schema later on, which is intimately related to PubMed, called the mesh database, which also does a job of capturing NAMs just in a different way. And the last part of this talk relates to literature searching, and there are many options here for navigating the literature landscape around NAMs. So there are curated collections and great literature sources out there. So that includes things like the inventory of 3R's knowledge sources, your ECVAM dataset on alternative methods, and the NAT database. There are at least one, there may be more, but at least one machine learning derived collection called SMAFIRA, where you can put in a PMID of a PubMed article, and it will, PubMed article, which is an in vivo model, and it will sort of in a machine learning backend algorithmic way post to you or present to you results that are alternatives to that in vivo model. And then there are bibliographic databases, which are not curated by topic, but are simply representing the output of research in biomedicine and other fields. And that includes PubMed, Embase, Web of Science, Scopus, and others. So these are all sources for accessing the literature. They all have their pros and cons. We're going to focus today on PubMed as a source of a great deal of the biomedical literature. And it's a tool that's freely available to everyone and represents a real core source of accessing the literature on NAMs. Any questions about kind of the laying the groundwork here for my talk? And again, you can put that in the Q&A if you have any. Okay, so like I mentioned, I'm going to talk about 5 processes that are key to developing comprehensive searches. And I'll just say a little bit about sort of the context here and the goal of this kind of searching. The goal is to not miss something that is critical to a research question. The goal is comprehensiveness in this context. And I would say that comprehensiveness is really a virtue in many settings. So particularly if you're doing an evidence synthesis of some kind, if you're doing a systematic review or a scoping review, for example, of course, you're required to develop a comprehensive search, multiple databases, and present that search in an appendix when you publish your paper. But there are many other contexts, I think, where a comprehensive search is valuable. That might be when you're doing a narrative review, or that might be when you're doing a grant proposal, or when you're trying to figure out what's been done in a particular area because you're wanting to try that yourself. So I think in many different contexts, the goal of not missing something that's relevant is there. And so that's the sort of endpoint of these processes that I'm going to go through. And the goal of implementing or making these implementing these processes is to ensure you're not missing something that might be relevant to a question you have. So there are 5 processes, and what we'll do, we'll work through each of them, what is the rationale behind them, and then talk about how you actually operationalize that process. And then we'll have some hands-on practice where we'll actually do that, or we'll actually put it into action. And the end goal here, as I mentioned, is for you to have a search at the end of the day that, while not complete, is nevertheless an exemplar of the kinds of approaches that are needed in order to build a search that is aspiring to comprehensiveness. And I'm just going to be checking this. Okay, awesome. So process 1 is to actually articulate a research question and identify its key elements. The rationale here, and also to some extent what I'll be talking about too, are habits of mind, things that searchers think about and orient themselves to, things that, in my experience, over 10 to 12 years of doing this kind of work, are sort of ingrained in me to think about. And so part of the goal of this presentation is to share those kinds of habits of mind and approaches, sort of the searcher mind, the mind of the searcher, as it were. So the rationale behind articulating a research question and identifying its key elements is to establish a conceptual framework for searching. And in the process, you're fostering precision. You're trying to focus your search and design your search such that what you get is really what you want. And you're limiting thereby the number of off-target things that you're getting. So this may seem like a trivial step, but in many ways, this can be quite challenging And it is, in any case, whether more or less challenging, is really critical because by establishing and identifying the key elements in your question, you are setting the groundwork for everything that follows in terms of the search that you're going to build. So this is the scaffold, as it were, the foundation for all the building and all the mechanics that are coming in later processes. So we're going to work from a sort of exploratory research question. How have organoids been applied to understanding or treating inflammatory bowel diseases? So this is a fairly unstructured search. It's really designed to see what is the landscape of the literature on this topic. And it's somewhat focused, but it's not as complex as research questions can get. But it has a few moving parts and not too many. And it's, I think, an appropriate one for this talk because of just sort of the time constraints we have. So for a question, for this particular research question, relevant studies or studies that are going to address this have to be about a couple of things. They have to be about organoids and they have to be about inflammatory bowel diseases. So again, this seems maybe sort of obvious and sort of trivial, but it is important to do this step and to really think about it. It's actually important to articulate your research question first of all, to actually have that front of mind and to know, what is your goal. And in addition, it is important to think hard about what are the elements of this question that are really important. So it's important to take a parsimonious approach here, to think what are the elements of this research question that are absolutely necessary for the results, to be relevant. And it's important to resist kind of sort of key element creep, as it were, to resist putting in or structuring your search around elements of your research question that are not absolutely critical. And so an example here, even with this short exploratory research question that has some pretty clear elements in it, you don't want to create concepts in your search related to implied concepts like applications, understanding, or treating. So, if someone's going to do research related to inflammatory bowel diseases and organoids, it sort of really is implied that they're doing that so they can understand things or that they can treat that disease, and that in effect, actually, organoids are an application. And so these are not really relevant or necessary key elements. And if you employ them, you run the risk of then excluding papers that are relevant. So it's important to think, what is it really that has to be in these articles to make them relevant? And so if we think about this a little bit more and visualize this a little bit more in terms of what our ultimate goal is, we can sort of take this structure and visualize it in a different way. So using terms just related to organoids gives me about 36,000 results in PubMed. Terms just related to inflammatory bowel diseases gives me about 160,000. And really what I'm looking for is that overlap of the universe of these two aspects of the literature. I'm looking for this set of relevant studies that include elements of both of these types of main ideas. So that's going to be around 600 studies. So this is our goal. This is our, what we're shooting for. And having a sort of well-structured research or search strategy is the way we're going to get to this goal. And so along the way, I'm going to sort of mock up a search, which will be sort of representative of a Word document or an Excel document or whatever tool that you might use to document things. Basically, what we're wanting to do here is create a kind of lab notebook for searching. So I like to express this as a series of experiments that we're running basically in PubMed or whatever database we're searching. And one of the processes we'll talk about later is iteration, and having a record of what you've searched, what you've tried, and a record of the success or the failure of that experiment. In other words, way too many results, way too few results, lots of off-target results, or lots of on-target results. That's going to help you iterate. So that will help you optimize, as we'll talk about later. And in these kinds of richly semantic tasks, like searching, things will come to you as you put terms together, as you look at results from those searches. And it's really helpful to have a place to record what you've done, so you know what you've done, and not do it all on the fly, but to do it in a sort of coordinated way, so you don't sort of forget what you did before, you're not repeating yourself, and also so you can make notes of what has worked and what hasn't worked, and what you want to bring to the next iteration of your search. So actually, at this point, what I'd love for you to do is open a Word document, which I'm going to do myself. And we can use that as the foundation for developing our sort of draft strategy. So I'm going to give this a title, organoids and inflammatory bowel diseases. If I can spell. So, and then what I'll do actually is take this actual research question and put it at the core of my, or right at the top of my document, so that it's, crystal clear to me what I'm actually searching for. And this actually is critical to know what you're searching for, but it's also critical to have there in case you may need to change your research question. So that's one of the things, and we'll talk about that when we're talking about search iteration. That's one of the things that can happen. You may do a draft search and find that you're overwhelmed with results, or that you're underwhelmed with results. It could be those two different, outcomes. In which case, maybe you really need to look at the research question that you started with to determine, is this manageable, or is this is the scope basically that I've established in the research question a good enough scope? Is it too narrow? Is it too broad? So having that there really helps to keep that front of mind. I'm going to call this first key element here that we've identified as organoids, right? That was one of our key elements. I'm calling it a concept, which is a sort of common parlance in searching. You tend to work with search concepts, and those search concepts are the places or the sites where you build search terms. And our second search concept is inflammatory bowel diseases. So now we have our framework. We have the main headings under which we're going to put our terms that correspond to those concepts. We're also going to put a notes field. And this is going to be a place where we can record any results from our experiments that we're doing. So bear with me as I do this. Okay. Now we have our search structure, basically, our framework, upon which we're going to hang terms and upon which we're going to develop our strategy. Any questions so far? So just going back to our slides, this is essentially what we've just done in our Word document. We've established this framework with our two main concepts or key elements, and then a notes field where we're going to record things like result numbers, relevance and noise in result sets, terms to remove and add. And we've finished our first process, which again is a critical one and something you may come back and revisit. So depending upon the nature of the searches you're getting, the nature of the results you're getting, additional concepts may need to be added to this to focus the search. The concept in and of itself could change to include maybe a specific inflammatory bowel disease, maybe Crohn's disease, or organoids might need to be expanded to other NAM approaches in order to find a critical mass of studies. But we'll talk about that in a later process related to search iteration. Now we're going to talk about a second process, which is really quite involved and represents, I think, a lot of the additional conceptual work of searching. So definitely establishing the framework is a key bit of conceptual work. This second approach is another important part of that, which involves systematically gathering the universe of search terms corresponding to these key elements. So that is a challenge. How does one systematically gather a universe of search terms? And I think this may bring us back to that anxiety we talked about initially, is like, how do I know where to get the terms? I mean, I'm familiar with terms that refer to these concepts, but where is a site I can go to systematically gather these terms? The rationale here behind this is to build a search that's comprehensive. So, you have to have the right terms. You have to, in a sense, give PubMed the instructions for what to find. And the way to do that, a key way to do that, is through terminology. Our goal here, and sort of habits of mind, is to try to think, how can I maximize recall? How can I make sure that I'm getting everything that might be relevant? And also, how can I be sensitive to synonyms, the many different ways that people refer to a particular idea, key element, or concept. So organoids are going to be captured not simply with one word, but rather with multiple terms. Same with inflammatory bowel diseases. And that's a real habit of mind of trying to think of how do researchers refer to these, and how can I capture that heterogeneity of terminology, and what source or sources can I use to capture that heterogeneity? And this is kind of based on an axiom that I sort of made up on my own, but I think it's a kind of truism, or it's not controversial, that basically within a particular search strategy, the more terms your search strategy has, the more results it will find, and by extension, the more relevant results it will find. So, I think the key here, and we'll talk about this later, is achieving a balance between recall and precision. So you don't want to obviously find tons of stuff that is not relevant. At the same time, you don't want to over-determine your search and find only relevant results. There needs to be some sort of middle ground there. And there is a sort of plateau that you reach when the addition of additional terms is actually not giving you more results. So these are all sort of speaking to that search iteration process, which we'll get to later. But at this point, really what we're doing is just trying to gather terms and make sure that we're capturing all the different ways that the key elements we've identified are expressed in the literature. And the way we're going to do that is we're going to use what's called the MESH database. MESH stands for medical subject headings, but it's a bit of a misnomer because it really refers or includes all of biomedicine. And it's a companion database to PubMed, and it's a place you go to find the terms that PubMed uses to describe articles. So it is a source of PubMed preferred terms. And just that sentence there is a clue to why it's valuable. So if this database of terms in the MESH database is the preferred terminology PubMed uses, then absolutely I want to be plugged into that, because that's going to be my key to finding relevant papers on the topics that I've chosen. It's also an engine for synonym generation. So it's a place where you can go to capture that heterogeneity I spoke about. So MESH terms, which are words or terminology that you find using the MESH database, are uniformly applied, representing these key ideas in biomedicine. And they're also sort of parts of, like in that NAMO ontological schema we saw earlier, part of this hierarchically structured system of terms. So within MESH, you're able to not only gather the terms that PubMed uses to describe its papers, but also see how those terms are related to other terms in a larger schema. And this is where I think it's a little challenging to capture everything related to NAMs. So in that NAMO ontological schema we saw earlier, that was developed specifically for NAMS practitioners and researchers, and really sort of was putting to the side everything else around biomedicine. The MESH database, which began in the 1960s, is designed to capture all of biomedicine. So NAMS coming along a bit later, in a sense, has to be somewhat shoehorned into this existing schema of all of biomedicine. And as a consequence, it's not as clean and not as sort of easy to visualize and to find as that NAMO schema, which was really fairly narrowly designed to reflect just NAMS research. And so as a consequence, the semantic landscape of NAMS in the MESH database is somewhat dispersed. So you've got, for instance, these grandparent terms in this hierarchy of terms, like investigative techniques. And within that, there are a lot of things that are investigative techniques, right? There's a lot of stuff that has nothing to do with NAMS that would be in this category, but there are things that relate to NAMS that are in this category, like animal use alternatives and in vitro techniques. So here we're seeing, particularly in vitro techniques, cell culture techniques, embryo culture, organ culture techniques that are hearkening back to that original ontological schema we saw earlier. So we're seeing that indeed NAMs are represented in MeSH, but you have to sort of hunt for them a little bit. And a couple of other examples of where NAMs terms appear is in completely separate trees, as it were, completely separate sort of organizational branches of MeSH. For example, there's a computing methodologies category, and underneath that are terms that are potentially relevant to NAMs, like patient-specific modeling or these molecular terms, but they're in a completely different area of MeSH than these investigative techniques. And then also there's a category related to tissues where we have organoids, and that's what we're going to look at today. So you can see there's a lot of dispersal, and there's not just one sort of place within MeSH where all the NAM stuff is going to be. So you have to be creative, and I think using that NAMO schema or some other schema that represents what NAMs are and what they include can be really helpful, because the MeSH database is searchable. So you can search for organoids and find where it lives. You can search for organ culture or tissue culture or cell lines, and you can see where those various elements live within MeSH, and then leverage that to use preferred terminology that's going to find papers that are relevant to those topics. So just to say a little bit more, organoids is, in this schema, unlike NAMO, a parent actually of microphysiological systems. In the NAMO schema, organoids was actually under 3D cell culture, and microphysiological system was its own category. So in this schema, in MeSH, they live in this relationship of parent-child. And when searching PubMed, while you can use NAMO to interrogate MeSH and figure out how MeSH organizes things, when searching PubMed, you want to be using the way that PubMed is articulating or visualizing NAMs. And so we'll be using organoids, and at the same time, we'll be capturing microphysiological systems as well. Because one feature of MeSH terms, which we'll look at in a second, is that they, when you use the parent term, you capture the child as well. So when we use the organoids MeSH term, we'll actually be capturing the microphysiological MeSH systems MeSH term as well. So it's a kind of you get two terms for the price of one, in a sense. So you're able, once you're able to kind of harvest two terms by using organoids, you're able to also incorporate the microphysiological systems term as well when searching. So let me pause there, actually, to see if there are any questions here about MeSH. Well, if you save them up, because we will also talk about them at the end. So if you have questions at the end, we can look at those. So again, just to reorient ourselves, we're using the MeSH database to identify these preferred terms. And we're going to do that because PubMed articles are assigned these terms. So the MeSH database and PubMed are companions. They influence or inform one another. And the whole reason for the MeSH database is to give what used to be human indexers and now machine indexers the codebook or the thesaurus or the set of terminology that is going to be used to describe papers. And those MeSH terms show up in the PubMed database in the records of individual PubMed articles. So maybe 10 to 20 MeSH terms are assigned to each PubMed article. And they appear actually in the PubMed record. And so let's take a look at sort of how that looks in PubMed itself. So MeSH terms are great. They're preferred terms. How do we how can we think about them as searching elements other than being preferred terms? Just to say, repeat myself a little bit, we're leveraging these uniformly applied descriptors. We're taking advantage of the fact that these terms are the ones that PubMed uses and applies across the board to any paper related to what we're interested in, and that we're tapping into PubMed's codebook essentially. And what we're doing when we use MeSH terms is we're targeting a particular field in PubMed. So basically, we're saying we're designing our search so that it hits on a particular part of the PubMed record. And when it hits there, it's going to retrieve it. So just a little bit about how the PubMed record is structured. It's structured as in parts or elements. And many of these parts, most of these parts are searchable. And these are referred to as fields. So there's a journal field, a journal name field, there's a publication date field, volume field. And that sort of appears in a PubMed record like this, where we have the journal name and when it was published and the DOI and so forth. There's another field called title, which is just the title of the article. There's another field or part of the record called abstract, which has a description of the paper. And then there's a section of most PubMed records, not all, and we'll talk about the significance of that in a second. But most PubMed records will have these MeSH terms assigned. So that's 10 to 20 of these MeSH terms that summarize what the paper's about. So we're going to be targeting our search to those parts of the PubMed record that are most important in summarizing what the content is about. So clearly title is important as a source of what is this article about. So is abstract. So is actually author-supplied keywords, which is not represented here, but is actually a searchable field. And then MeSH terms. MeSH terms are basically summarizing a list of summarizing terms for this article. And it's important to think about these MeSH terms and what we'll later talk about, which are keywords, as sort of separate entities that have separate functions. They are synonyms of one another, but they have different targets within the PubMed record. And so the MeSH terms are targeting the MeSH field. And when we'll talk later about keywords, those are going to be targeting the other really richly semantic summarizing content of a PubMed record, which is the title and the abstract. So the MeSH terms that we are putting into our search are going to be targeting this part of the PubMed record. So let's do some hands-on practice, which will involve gathering MeSH terms that correspond to these key elements. And the rationale here, and the way we're going to do that is by searching the MeSH database. And the rationale here is to include these preferred terms in our search. And also to think about and see these terms as part of a system, as part of parent-child relationships. So let's do that now. So I'm going to go into PubMed. So you can do the same. Oops. So if you just Google PubMed, takes us here. And down below PubMed is a explore section, and there's a link here to the mesh database. And you could also just Google the mesh database if you wanted to. And so this is the tool we're going to use to find preferred terms. One way to think about it is that when you search PubMed, you're finding articles. That's the results. When you search mesh, terms are going to be your result set. And so really what we're doing here is just trying to see, do the concepts or main ideas that we've identified correspond to preferred terminology? And is it exactly the same term that we thought of, or is it slightly different? How does it map, basically? So we're going to do our first concept, which is organoids. And so we type that in and we search it, and we see we get a couple of results. And actually, what's interesting is we get the parent and the child here in these results. When that happens, we can just go to the parent term. So this is the mesh record, and we'll talk a little bit about how this is structured. It's also structured by fields. We'll talk about that a little bit later. But basically, this gives you a definition of what PubMed thinks organoids refer to. So you can look at that and make sure you agree before you actually use the term. And then the other thing I want to point out here about this record is the hierarchy down here. This is the kind of ontological schema here down below. So it's part of tissues, and it has a child term of microphysiological systems. So when we use the organoids mesh term, we're going to capture the microphysiological systems mesh term as well. So how do we do that? We can come up here to the PubMed search builder on the right-hand side of this record and click on Add to Search Builder. So now what we have here is a fully formatted mesh term called organoids. And what we can do now is just copy it and paste it into our Word document. So this is our first term for our first concept. So we're building right now. You may have thought we were never going to get to this stage, but we're here now. And now we're going to do inflammatory bowel diseases. We're going to see, is there a mesh term that will search the mesh part of the PubMed record that corresponds to inflammatory bowel diseases? And so we're just going to run this here and see. And in fact, we get a single hit. And so it takes us directly to the inflammatory bowel diseases record. And it shows us that there are children terms. So there's ulcerative colitis and Crohn's disease that are included underneath this inflammatory bowel disease parent. So we know when we use this parent mesh term, we're going to capture these mesh terms as well. What I will say now is that keywords, thinking about keywords, which is going to search a different part of the PubMed record, we're going to need to gather keywords from these child terms. And we'll talk about how to do that systematically in a second. But for now, what we'll do is just capture this mesh term, which is really really an awesome term, because it's going to capture not only this sort of main idea of inflammatory bowel diseases, but also these specific diseases underneath it. So this is a really nice term to incorporate into our search to make it more comprehensive. And we just plug it in right here. So now we've done our work related to gathering mesh terms, and that is the first part of process 2. But just going back to our PowerPoint, there's actually a second part to process 2. But before we get to that, let's just sort of see what our mockup looks like. Again, 2 concepts, 2 mesh terms. And I will say at this point that actually concepts can be represented by multiple mesh terms. So we just have a sort of one-to-one here, but there are cases where, particularly say our concept 1 was not organoids, but was NAMs writ large. In that case, organoids would be a mesh term we would want to include. but we would probably also want to include mesh terms related to various in vitro techniques, or mesh terms related to in silico models, and potentially mesh terms related to animal use alternatives. So mesh terms can be, or there can be multiple mesh terms that correspond to an individual concept. Great. Any questions about what we've done so far in terms of search building?
There are some questions in the chat.
Oh, awesome.
For example, there's a general question, if you want to hear that now, best.
Yes. Yes, let's do it.
Do you have any tips for obtaining literature that will not be found in PubMed? That means those published in journals not indexed by the National Library of Medicine's Medline database.
That's a great question. And it's a question particularly relevant to certain evidence synthesis projects like systematic reviews and scoping reviews. You have to search multiple databases in those contexts. The reason is because no single database is going to have in it all of the published literature. So there are choices that PubMed and other databases make in terms of their scope. And we'll actually address this a little bit when we talk about keywords and why they're important. So PubMed will say, well, these particular journals, while they're fine and all, but they're just sort of outside of our scope, so we're not going to include them. We're not going to index them. We're not going to take in the articles from these journals. A database like Embase, which actually includes much of what's in PubMed and more, is going to cover more of the literature, say, related to European journals or related to drugs, for example, and other topics. So they've made a choice to incorporate a different set of journals, which will be, to a large extent, duplicative or redundant in terms of, say, PubMed, but will also be unique. So when you want to do something really comprehensive, and this is getting sort of outside the bounds of PubMed, you would translate your PubMed search that you develop into, say, an Embase search, or even a Web of Science or Scopus search, which also will contain uniquenesses in the coverage of the literature. And then you'll have a step where you remove all the duplicates. And there will be many duplicates across all these different databases. But in general, the tip would be to, if you're wanting to be absolutely comprehensive, would be to search multiple sources. And certainly databases that come to mind that I have definitely used in evidence synthesis work is Embase and also Scopus or Web of Science. Scopus and Web of Science are multidisciplinary. They cover biomedicine, but also lots of other topics. So they're great for sort of multidisciplinary questions, but also great for biomedicine as well. So yeah, that's my tip there.
And then just a follow-up question on the mesh terms from PubMed. Do other literature search databases also use those or not?
Wow, you all are asking some awesome questions. And yeah, the answer is yes. Of the databases I just mentioned, Embase is another database that has what's called a controlled vocabulary. So I'm so glad we're talking about this because I can introduce the concept of controlled vocabulary. A controlled vocabulary, as the name suggests, is one which is highly rigorous and uniform. So this is going back to the uniform application of terms. It's highly structured in contrast to the heterogeneity and uncontrolled nature of how people talk about things just in natural language. Someone might refer to an organoid, someone might refer to a collonoid, someone might refer to these things in various different ways. As we all know, ideas in biomedicine are represented by multiple and sometimes a dizzying number of ways. And so MESH is a controlled vocabulary. in the sense that it is actually consisting of umbrella terms to capture that heterogeneity that's out there in the literature. And Embase is a similar, or I'm sorry, Emtree, sorry, is a similar controlled vocabulary. Emtree is what is the companion database to Embase. And so Emtree, just like Mesh, is searchable, and you would search it to determine what terms are used in Embase to describe papers related to my topic. Sometimes these terms are exactly the same across Mesh and Emtree, but sometimes there are important differences, and sometimes the hierarchical structures are quite different. And also, sometimes the synonyms that you will find when we get to keywords are quite different and more robust. So, excellent question. That whenever you're searching a database, a general rule of thumb is to look for something called a thesaurus or a controlled vocabulary that is a companion to the database, because that's going to be the source of the preferred terminology that's going to be helping you to be comprehensive in your searching.
Maybe one more question before we continue. There's a
Yeah,
question about the snowball searching. How and
yeah.
when to apply snowball searching?
Yeah, so snowball searching, as I'm understanding it, would be a case where you have, it's also maybe referred to as citation searching, where you have a on-target article and you want to see what other papers have cited it, and potentially also going backwards in time, what papers did it cite. Um, and I think that is a helpful approach. Um, an alternative approach. It is, I would say, narrower than a narrower approach than a concept-based approach, which is what we're doing right now. And I think also there's, I think there's definitely an issue with signal to noise ratio in a concept-based search, which we'll talk about a little bit. But also there's that in a sort of snowballing or citation-based thing as well, because not everyone who's citing this paper will be citing it for the reasons you want, right? Sometimes they'll just cite it as background for something else. In other words, not all the papers that cite a paper are going to be relevant to your question. But it can be quite a useful approach. And I would suggest, anyway, I think it can be used in multiple ways. I'll just say it can be used in and of itself as an approach. Um, so knowing kind of the caveats of, the limitations of the approach going in, I think is useful to know that there are some limitations on what you'll actually be able to identify, and that there will be definitely things that you will not find that are relevant because of the narrowness of this approach. But it can be used on its own. It can also be used, I think, helpfully as an exploratory exercise to kind of acquaint yourself with the landscape, and to also get a set of on-target articles, which we will talk about near the end, about search validation and how to know when to stop and that kind of thing. So this kind of snowballing or citation searching can help you identify a lot of relevant papers, which then can become a gold standard or on-target set, which can then be used to validate your concept search. So it could be used in conjunction with a concept-based search as a way to generate this kind of gold standard set, which can then validate what you create when you build with concepts. Hopefully that makes sense. Okay, so let's continue on. We're again, oh, and I will say this is an opportunity to just note what I said before, that you're going to want to include keywords for child mesh terms. So there are a number of children in this scenario. Microphysiological systems as a child of organoids, ulcerative colitis, and Crohn's disease are children of inflammatory bowel diseases. And so we're going to want to make sure that we fully integrate the terminology that we can get from mesh records in the rest of our search. So that brings us to keywords. And I'll explain more what I mean by this. And we'll actually do it when we continue building our search and incorporate keywords. So keywords for searching. Why do we need keywords when we have these preferred terms from mesh, you might ask? I would say it's really kind of going back to the scope of a database. So PubMed does not index or describe with mesh terms every paper that is in it. And I think part of this is based on the way PubMed is scoping. It says certain journals it wants to include, but these journals are maybe a little bit out of scope, a little bit tangential, and so they're not going to take the time to index those, possibly because they don't feel they have the right mesh terms to describe them. So there's some sort of back-end decisions about which journals to fully index and which not to. And so there are some records or some papers in PubMed, I don't know the percentages, but a fair number that just don't have mesh terms at all. And so if you just search with mesh terms, if we just ran the search that we just built, we're going to miss a lot of papers because there'll be papers that just won't hit. There won't be any hits on them because there are no things to hit on. There's no mesh records to hit on. So that's one thing. Also, mistakes are made. So, these machine tools that are now being used primarily with some human oversight to index terms in PubMed can make mistakes, and mesh terms that maybe should be applied to an article aren't applied to an article. So keywords can be thought of as backups, and really critical backups, as we'll see in a second when we look sort of at what is found by keywords versus mesh terms. They're backups to mesh terms to make sure that you're including all those papers that don't have mesh terms, and you're including all those papers that maybe should have those mesh terms but don't for whatever reason. And this keywords can also be thought of as capturing this terminological heterogeneity. So there's many different ways of referring to the same thing in the literature. So it's a little bit of a paradox maybe, or a little bit counterintuitive, that we're using mesh terms, which are umbrella terms, which are designed to capture all the terminological heterogeneity. So if someone refers to it as an organoid, or if someone refers to it as a colloid, it's still going to get the mesh term organoids, right? That's what a mesh term is designed to do. So it's a little bit maybe counterintuitive that you want to include both those terms that capture the heterogeneity in an umbrella way, and also all the individual terms as well. But in fact, that's what needs to be done because it's an imperfect system, because it's language, because language is inherently complex. We just have to include all this variety and different ways. Again, thinking broadly with synonym sensitivity, how do people talk about these things? And so we have to include keywords in order to incorporate that heterogeneity. So looking at the PubMed record and the fields that these terms, again, these keyword terms, which are related to and sort of synonyms of mesh terms, but are distinct in how they operate. And the distinction here is that they're going to search a different set of fields or a different set of the parts of the PubMed record than the mesh terms are. So going back to our PubMed record, we have the same fields, but really title is what keywords are going to hit on, and abstract. So this rich descriptive content is what we're using keywords to hit on. And also what's not indicated here, author supplied keywords, which are sort of the author's mesh terms in a sense. The author says, my article is about these 4, 5, or 6 key things. And they'll list it, down at the bottom of the abstract. So the keyword searching that we're going to do is going to hit on those parts of the PubMed record, and the mesh terms are really not part of this, at least in how we're going to do keywords today. We're going to use a field that focuses just on the title, the abstract, and the author supplied keywords. Okay, so just to make the case quickly about why keywords are needed is going back to that sort of initial Venn diagram where we were talking about organoids, the universe of organoid studies and the universe of inflammatory bowel disease studies and the intersection, which was the goal of all this work. Just looking at the organoid study part of that universe of literature, that was about 36,000. So from mesh terms, when we do a mesh term search, so that would be the organoid mesh term, basically, which also captures microphysiological systems as a mesh term, we get about 17,000 of these 36,000. When we use keywords that correspond to these organoid studies, so that's going to be keywords related to microphysiological systems, which actually turns out to be like organ on chip, organoid model, organotypic model, that kind of thing. We get 29,000 results. So there's an overlap here of about 9,000 papers. And then there is uniqueness. So 8,000 of these papers were only found by mesh terms, and 20,000 were only found by keywords. So you can see, hopefully, why both of these kinds of terms are really important. So where are we going to get keywords? So keywords come from many different sources. One of them is your head, right? Or your experience, or your knowledge, or your expertise. You just know terms of art referring to concepts. If you're an expert, if you're not an expert, then maybe you'll rely on different sources. And those sources might be key papers, or they might be book chapters about NAMs or something like that, where you start to harvest terms and incorporate them then as keywords in your search. That's a little, that's somewhat ad hoc, but really not necessarily. I mean, if you sort of go to a core paper or go to a core definitive book chapter, that's pretty systematic. Like you read those and you say, okay, these are trusted sources. I'm going to gather terms from there. That is part of a system that's logical and fairly comprehensive and systematic of gathering terms. Another approach, and all of these approaches actually can work together. It's not an either or, it's like all of the above, is to use those same mesh records where we got the mesh terms. So we're going to focus on a single source for systematically gathering terms today, which is the mesh database, and specifically the mesh record that corresponds to what we're interested in. And so how do we do that? We're going to harvest terms from what is called the entry term list in the individual mesh records that we've already visited. And so this is a little mock-up of a mesh record. So similar to a PubMed record, it has different parts. It has a mesh term field, which is, in this case, microphysiological systems. It has a scope note, that definition I referred to earlier, which basically says, this is our understanding. This is PubMed's understanding of what microphysiological systems are. And I will say that the mesh database has some linking elements. It has some see also references, and it also has this all caps linking device. So basically you see all caps in the scope notes. That means that there is a corresponding mesh term related to microfabrication, microfluidics, and 3D cell culture. So this is also a kind of way that you can get, oh, I should go to that place and I should incorporate that mesh term as well. Okay, so that's just a little aside. So there's a scope note, which is the definition. And then there are what are called text words in the mesh record, which is also called entry terms, which is what you'll see, and we'll see this in a minute when we go into a mesh record. This is what the heading is called, entry terms. And that is a whole list of keyword instances that appear in the literature that are things that are used by researchers to refer to this concept that's represented in mesh. So people talk about the singular microphysiological system. They also talk about organs on chips, organotypic models, organ chips, and a whole list of others. The ellipsis indicates that there are more terms. So this is really a great source for keywords that you may already be familiar with, or maybe terms that just you would never have thought of. Again, this isn't exhaustive. I want to stress that, but it is represents, I think, a systematic approach in the sense that you can say, well, I built this search by gathering the mesh terms for these concepts, and then I went into the mesh records and I gathered the keywords that I know exist in the literature as instances of how this is expressed. So that is a system and is defensible and is a foundation and a ground upon which one can build a confidently quality search, I would say. But having said that, it's not necessarily the be-all end-all. There are other sources, as I've mentioned, for keywords that you can also include and add to. And one of those sources actually is the results themselves. So we'll be talking about a feedback loop in a second, where the results actually improve and help you optimize additional versions of an initial search. Okay, so this is all a little bit theoretical. So how do we make this real? So let's do some hands-on practice and gather keywords corresponding to key elements from mesh records. I will say, though, going back here, that I've done a little editing. Entry term lists are awesome, but they're also flawed. So they do have some weird last name, first name formats. And what's important to think about here is the field, the target. Where are you sending these keywords to hit on papers in PubMed? And knowing that it is the title and the abstract and the author supplied keywords, you know, oh, well, I don't want to put them in last name, first name format. People don't write that they used 6 different models, organotypic. They say we use 6 different organotypic models. So I did a little bit of editing of this entry term list to flip those terms around. So there is a fair amount of redundancy in this entry term list, but knowing where your terms are hitting can help you navigate that and not include terms that are in this weird sort of inverted form. Okay, so we're going to go to the mesh record, and I think this will be clear when we look at it. And the rationale here is to supplement this preferred terminology from PubMed and build comprehensiveness at the same time, as we saw from this Venn diagram of the contribution of these types of terms to our results. So let's do that. We're going to do that by going into the same records we've already been to, honestly. From mesh, we're going to go to the advanced search, which shows a history of what we've done. So we did a search on organoids. We did a search on inflammatory bowel diseases. We're going to just start with organoids because that's where we began. Click on the number 2 to get back to our results. And we're going to go to organoids. We're not going to go into the entry term list for microphysiological systems, just in the interest of time. But we are going to grab this entry term. So the entry term here is very short. It's one term, and that is not the norm. Normally, there are like 10 to 15 entry terms, but this has one. So we just copy it, basically. And then we're going to paste it into our Word document. We're going to leave a little space or a little marker here. And we're going to paste it as like plain text so we don't get the bullet. And that's it. But then we also know English, right? And we know that there are plural forms. So some databases will search singular and plural forms. PubMed is not one of them. So we're going to incorporate the plural as well. So we have now created a string, or at least a list of terms related to organoids. We're just going to choose two, basically, two keywords. And now, in a somewhat more complicated scenario, we're going to look at inflammatory bowel diseases. This also has one entry term, really. Here's that inverted form, which is basically the same as this. So we don't want it because it's in a weird form, but we do want the singular of it and we want the plural of it. So we're going to capture this one and put that into our Word document. Again, leaving some kind of, I'm just using a little space bar or whatever this is called, a little separator. And I'm going to put the singular, just like I did with organoids, and then the plural. And this may all seem kind of trivial and like, of course, duh. But I think we'll see as we're building this out, how we get some really unique terms we may never have thought of. And that is going to be when we're looking at the child terms. And that's where the complexity of entry terms gets a little more real. So as I mentioned, mesh terms and keyword terms are searching different parts of the record. So it's good to think of them as distinct. And so we always want to think of the child terms and not leave them out. We want to make sure we look for keywords in the child terms as well. So we're going to open up ulcerative colitis. So you'll notice actually this is colitis, commas, ulcerative, and that's fine for a mesh term, but not for a keyword. And if we go down to the entry term list, we see these things that are starting to be more different. And you're starting to see, I think, the value of entry terms in generating keywords you may not have thought of. So we're going to grab just the ulcerative colitis term as well as this one. And that's what we'll do. We're just going to leave it there. But we're going to know that there are additional terms like colitis gravis and idiopathic proctocolitis that we should also be including. But we're just going to do this quick and dirty draft right now. So this is what our search looks like right now. Any questions about this keyword term harvesting? Awesome. Okay, so we have sort of completed a really key part. Process 2 is pretty in-depth. And process 2, in addition to process 1, is really conceptual and key, really key to map out the framework and then to see where that framework maps to this structured ontological system, which is the back end and the underpinning of PubMed. So we've done that. Hooray for us. But there's more to go. And these next processes are going to involve, at least the next two, are going to involve more mechanical steps, but are equally important. And actually, we'll go quicker. So we are, I think, at like 40 minutes or so. So these next steps will go much quicker. But let's see where our search mockup is. Again, we've got organoids, and this is a more thoroughgoing harvesting of terms. But even with the ellipsis, you see it's not complete. So these are all the terms related to organoids, including terms related to the child term of microphysiological systems. So you're seeing all those organotypic model, organ chip, all that kind of stuff there. And then the same for inflammatory bowel diseases, which includes ulcerative colitis, but also keywords related to the child term of Crohn's disease are also included here. Okay, process 3 is uniformly formatting all of the search terms based on the search target. That's already done for our mesh terms. If we recall, mesh terms, when we use the PubMed search builder, have quotes around them and have a bracketed field tag. So I haven't mentioned that. Forgive me, but that is the instruction of how PubMed should search that term. So organoids in quotes with a bracket mesh after it tells PubMed to look in the mesh part of the PubMed record for the word organoids. That's how the targeting happens, basically, is with these fields. And we'll talk a little bit more about that in a second. So we're going to base that on the search target. So for keywords, we're going to use a different tag, a different bracketed expression to target those keywords to particular parts of the PubMed record. The reason we're doing this is to ensure that the terms are searched strategically, just as we want them, and to foster precision. So I will say right now, something I haven't said before, which is that all fields is the default. For if you just put terms into PubMed, it's going to search every part of the record. And that could be problematic. If your term is hitting in the journal name, potentially in the author name, potentially in author affiliation, that could be good, but it also could be really off target. So we're taking the approach of strategically telling PubMed just where we want it to search to avoid these off-target possibilities. Okay, so we're going to do that through field tags, as I mentioned. And the PubMed user guide, which I linked to here, has a lot of great information about searching, not only with field tags, but with everything about searching PubMed. So I recommend you take a look at that. And we're going to apply field tags and we're going to apply quotation marks to our keywords. Again, these are already assigned to our mesh terms. We don't need to do anything with those. But in order to strategically search with keywords, we need to do these two steps. And so field tags are many in PubMed, many ways to focus your search to particular parts of the PubMed record. The all fields tag is represented here in red in this first column, but you can see others like affiliation. If you want to search for a particular institution or a particular author, you can designate that and focus your search. And so the convention here is basically you put the term in quotes, and then after it, you put whatever part of the record you want it to search in. And this gives you the code to do that. So author would just be bracket AU, close bracket. There's another category here of field tags, including mesh terms. So we see here an example of something where there are sort of redundant or multiple ways of expressing the same field tag. So we are using mesh, M-E-S-H in bracket, but you could also use MH as an abbreviation in brackets. So both of those will achieve the same end. And then the last column includes what we're going to be using for keywords, which is TIAB. That searches the title, the abstract, and author-supplied keywords. Okay, so how do we make this work? How do we tell PubMed where we want our keywords to be searched? We're going to do that now with the TIAB field tag and quotation marks. And the rationale, again, is to dictate to PubMed exactly how to handle terms. We don't want PubMed to do anything to map things, and it has a feature called automatic term mapping. If you put a single word in and PubMed doesn't find it, sometimes it will map it to something it thinks you mean, which could be sort of catastrophic for your search. So that's why even for single terms, I put things in quotes. It can also potentially split up a phrase into its component parts and them together, which also could be fine, but also could be problematic too. If one of the words is a commonly used word that has many contexts, that could end up giving you a bunch of off-target things. So to foster thoroughness, we're going to just say, put things in quotes and apply field tags. And it's really important to be thorough here. If you leave out a quotation mark, if a quotation mark is unclosed, then that can wreak havoc on your search. So these steps are more mechanical, more rote, maybe, but they're equally important in terms of the running of the search. So you have to really be thorough in these steps. So let's do our hands-on activity here. Going back to our Word document, it's really quite straightforward. We just put quotes around these terms, and then after the quote, we don't even need a space. We put the bracket TIAB, close bracket, to indicate where we want this to search. In this case, the title, abstract, author supplied keyword. So at this point, it's a really bit of a somewhat of a slog. Not really. And I've kind of used various techniques in like Excel to kind of automate this. There are probably other ways to automate this. Maybe, ChatGPT can automate this too, even. But so it's fairly quick though. Basically, we're just applying these quotation marks and doing the field tag, making sure that we're not making mistakes. And there we go. So now we have keywords that have the same structure as our mesh term. So again, the mesh term in quotes, mesh in a field tag to indicate to search in the mesh part of the record. Here's an example, ulcerative colitis as a keyword in quotes with TIAB to indicate where it should be searched. So we're making progress, which is good because we have 30 minutes to go and we have a fair amount to cover. So any questions though about what we've done here with our field tagging? Okay, awesome. So in terms of our mockup, we've got this. Again, our keywords are fully formatted now with quotation marks and field tags. So we're really getting close to having strings we can run. But as I mentioned, we don't have search logic. So we need to talk about that as process 4. Essentially, we want to specify exactly how terms will be combined. And that's important because if you dump in a bunch of terms in PubMed, it will just put ands between every term. So it will put the Boolean operator and will assume that you want the universe of these individual terms, how they overlap. Or basically said another way, PubMed will assume that you want papers that have all of these terms in them and must have all of these terms in them, which is not always the case, right? You're building a string of synonyms under these concepts. And basically all of those could be things that are in the paper, but they don't all have to be in the paper. Basically, you're just looking for representatives of terms from individual concepts, and it could be any combination of those terms. So we want to be clear with PubMed about how to combine terms, and we don't want to leave it up to PubMed because PubMed will probably make a mistake of how we want to combine these terms because we're building strategies that are complex. And so it's not going to be a simple term. One term represents one concept, one term represents another concept. I can put them both in and the and is fine because that's what I want. That's not what's happening. We're building in strings of synonyms. So we need to be sophisticated about how we specify search logic. And we want to be thinking about how do we assemble these moving parts. And so to represent the overall search strategy Venn diagram, we have this set of terms related to organoids, and we want to use or, the Boolean operator or, to signal to PubMed that any of the terms in this list of synonyms is fine. They all don't have to be in the paper, just one of them, any one of them in the paper is great. Please pull that down for me as a result. And so that looks like this, term 1 or term 2 or term 3. Maybe this is kind of what that question also earlier was asking about. So this is the general structure here. The same is true for inflammatory bowel diseases. We want to use or to connect those synonyms together, term 1 or term 2 or term 3. And then as a culminating step, what we're interested in is the relevant studies and how we're going to achieve that as with the Boolean and. So that's going to show the intersection between these two universes of the literature, where they intersect. So how do we do that? We're going to use these operators. We're going to use or within concepts to connect synonyms, and we're going to use and between the concepts. And the way we're going to use the anding is we're going to represent the long string of terms using a kind of placeholder. We're going to call it basically concept one. And then we're going to say we're going to and concept one to concept two. And we're going to do this sort of simple kind of representational approach to avoid errors. And we'll talk about that when we actually build the search and run it. So the rationale here, again, is to dictate exactly how to combine terms. And we have to have attention to detail. We don't want to leave out an or, for example, or an and will get inserted. So in a long line of synonyms, if you put an and in the middle, that's telling you PubMed something quite different than if it's an or. Basically, it's saying you need to have multiple synonyms in a record before it will get pulled. And that's not what we want. We're interested in any instance, and it doesn't matter what term. They don't have to be together. At least the synonyms don't have to be together. Okay, so let's do that. So this is very quick. We're going to go back to our Word document and just replace either the space you put in or whatever placeholder you put in between terms. We're going to put the or within a concept. Okay, so that's really easy. And then the same down below. So yeah, it's basically saying, PubMed, I want this, or this, or this, or this, or this. It doesn't matter what term. And then we have this culminating expression, which I have yet to write, but it's here. Concept one and concept two. This is done this way as a way to avoid possible error. As we get super long strings of terms, say super long, but as we get, say, like a paragraph length of terms, we want to be careful about combining it with another paragraph long string of terms. And we're going to use the advanced search builder to do that in PubMed to avoid errors of parentheses. Because when you are combining these, sort of like in a math or arithmetic expression, you want to put things in sort of order of operations. You want to or the things first, and then you want to and them to the other or thing. So you use parentheses to do that. But I discourage people from doing those things manually. So we'll get to that in a second. Okay, so any questions about the Boolean operator application? Awesome. Okay, so now this looks like this. Basically, the ors are there within the concepts. And then you have the summative statement with the and in the middle. And we are nearly there. We're almost there to process 5, where we are going to do some search iteration and validation. This is an important step. The rationale here being is that your initial search is almost never the final search, at least in a context where you're shooting toward comprehensiveness. I'm not saying that's always the case, right? Sometimes a quick and dirty search is all you need just to find some papers and maybe a review article, right? But if the goal is comprehensiveness, then your first search is almost never going to be your final search. And you want to optimize that. And you also want, in this kind of potentially anxiety-ridden state, when can I stop? When can I stop iterating? When can I stop optimizing? And the way we're going to answer this and the way to think about it, I think that's helpful, going back to this sort of Word document as lab notebook, is through experimentation. So we're going to do a series of experiments here. And through those experiments, we're going to learn various things, and we're going to incorporate that learning into our iteration. And so one way to think about it is as a feedback loop here, where certainly our search strategy is informing our search results. We get no search results if we have no search strategy, right? So definitely you see that connection, but there's also this feedback from the search results informing the structure of the search strategy or informing the optimization of the search strategy. So an example might be you get some on-target results. Yay, from your search strategy. You can actually leverage that by looking at those results fairly carefully and potentially identifying terms that you are seeing in those search results that you hadn't incorporated into your search yet. So, for example, you might see an article that talks about colloids, which you don't have in your search string, but could be really valuable to generate other results that only use colloid to talk about an intestinal organoid that is used to study inflammatory bowel diseases. Another example might be you get too few results. And one approach to address that in your search strategy might be to broaden your concept. So you might consider broadening out the diseases you're looking at beyond inflammatory bowel diseases to maybe a family of broader diseases that include them, including gastroenteritis disorders, which is a broader category. You might get off-target results, which is very helpful. Well, sort of not super welcome, but helpful in optimizing your search strategy, because you might see trends in terms of the terms in the title, the abstract, or even mesh terms that are generating those off-target results. And so you might want to remove those terms to avoid getting those off-target results. So an example might be an abbreviation for IBD, which is a fairly common abbreviation for inflammatory bowel diseases, but is also an abbreviation for iliac branch device. And so you might want to exclude IBD. And actually, when you exclude a term like this, or even when you add a term, you can do an experiment where you see what is the impact. If I take out IBD, do I then not get something relevant? And maybe I'm okay with a few iliac branch device articles. I'll just ignore them in order to capture the IBD relevant ones. So there's an opportunity for experimentation there. Maybe you get too many results, which I think is maybe a fairly common situation. You might want to add a concept, or one approach to this result might be to add a concept, say features of inflammatory bowel diseases or symptoms of inflammatory bowel diseases as a concept that you would combine with the other two. Okay, so let's talk a little bit about markers of sufficiency. So this gets at that kind of when can I stop? How much iteration is enough iteration? And also gets at this kind of ground or this foundation of quality that I've been talking about, of like, hey, I have done due diligence. And if I need, sometimes in, say, a systematic or scoping review context, you need to sort of justify this or articulate this directly. But even just articulating it to yourself, that I think I've done a good job here. Some markers of that sufficiency in search quality could be that you've included relevant mesh terms and keywords, employing the methods that we've discussed today. So if there are mesh terms that you're seeing in some of your search results that you don't have included and seem really on target, those should be included. But if you are not seeing those kinds of things, if you've searched the mesh database and aren't really finding any other terms, and if you've also harvested the keywords from those mesh records, you have a good sense that you've done a good job building your search. Also, markers of sufficiency, or maybe in this case, insufficiency, is whether or not you have a good balance of recall and precision and results. So you want to be, there's no hard and fast rule here, but you want to be somewhere between 1% relevant, right? Or you got like 99% irrelevant stuff, a ton of noise. You want to be somewhere between that extreme and the extreme of 100% relevance, where you've probably overdetermined your search and maybe added like unnecessary concepts that focus it down overly much. And again, this is within the concept, or within the context, sorry, of a comprehensive search. An overdetermined search where you get 100% relevant could be quite appropriate in certain contexts, but I would say not in a context where you're searching for comprehensiveness. So you want to be somewhere between these two um, extremes. And then finally, and this is going back to that snowballing search or that citation searching, having a predetermined set of on-target articles before you even start searching that you can then use as a validation tool at the end to basically say, if I'm not finding these on-target articles, something's wrong with my search and I need to adjust it in order to find those. And that's where experimentation comes in as well, where you try various combination of terms to see, can I then get these on-target results in my results while avoiding, the pitfall of generating a bunch of noise in the process. Okay, so let's do some hands-on practice of basically using the advanced search builder. And we've got about 15 minutes here, so I think we have time. I'm sure we have time. This is really this is the finish line here, right? So we're going to run this full search in PubMed using the advanced search interface, and we're going to do it. The rationale here is to test search quality and to look for errors, and also to perform assembly. So this is all about accuracy in terms of a sort of habit of mind. I recommend not dumping the full search in for two reasons. One, it's hard to identify errors. It's easier to identify, or I'll say it's easier to identify errors in a search concept than it is in the full strategy. So using an advanced search builder in PubMed enables you to sort of get some ballpark numbers and see if those numbers make sense. It also will show you if you misspelled a word or left out a Boolean operator. So it's really a place where you can control for quality. And so the way we do that is search by individual concept, so we can interrogate that individual concept. And then the assembly piece is also a place where errors can occur. And that's why we're going to use the advanced search builder to do the assembly. Basically, to put the parentheses in the right place, put the and Boolean operator in the right place, and get our final result. Okay, so let's do that. So we're going to go back to our Word document. So these strings are ready. They are search ready. They have all the elements they need. Now we just need to go into PubMed. So if you want to Google PubMed or are already there, we're going to use the advanced search tool. So there's a tiny little link underneath the search box that takes us to the advanced search builder, which has a way for you to build a term at a time. But you don't need to do that. We've already done that. We built our search offline in our Word document, so we can put everything in the query box. And so what we're going to do is build it concept at a time. So we're going to take this organoid concept first, copy it, paste it into the query box, just like that. I like to just check spelling because it's annoying. So this is what our first search string looks like. And instead of searching, we're going to click on the down arrow and add to history. So there's a nice little history down here that shows us what we've done. And this is where you can sort of get a ballpark sense. 30,000 results for organoid studies. Does that make sense? I don't know. I'm not a subject matter expert, but it seems reasonable. If you got 30 or if you got 300, that would be a warning sign that something's wrong, right? And usually it would mean that you left out a Boolean operator or something, or misspelled a word, right? So if you look at the search details, this little caret here, it shows you exactly how PubMed interpreted your result or your terms. So it took your organoids mesh term and basically used it as is. So here's another iteration of how you can express mesh as a field tag, mesh terms. And then organoid was translated into a title and abstract term, just like you said. And so was the singular and plural. So you basically, you see here in the search details that you made no mistakes, and PubMed knows what you mean and is performing the search as you intended. Now we can go and take the second concept, add that to the history as well. We get more results, which is what we would expect, I think, given that there are so many diseases here, and not seeing any errors. We're seeing the same kind of one-to-one correspondence of terms to what I intend. So this all looks great. And now we can do the search building, right? So we have our components, but we have not combined them in our culminating step to see the intersection of these two slices of the literature. So we're going to do that using the action feature. We're going to add the first query by clicking on Add Query. So now that's in the query box. And then we're going to go to Actions for Concept 2 and use this Add with And action. And you see it puts parentheses around the organoid concept and around the inflammatory bowel disease concept, and then ands them together. If you had left out a parentheses, if you'd done this manually, that would give you a completely different result set and not the one you want. So this is now fully ready to go. We can add this to the history, and we get around 582, which is what we were expecting, that 600 number. That's what we were expecting. If we click on this result link, we can now do that sort of feedback loop step, right? Where we look at the papers that we're seeing in our results, and we're seeing, are they on target? Are they off target? Are there terms that they're using that are unique and things that I hadn't thought of? And one way we can help do this is if I'm logged in. So I'm going to log into my NCBI account. Bear with me for a second. I really recommend these accounts because one thing you can do with them is you can set up highlighting, and that your terms are then highlighted in your result set. So I'm just going to quickly get back to my result set. And I see the time is ticking away, so I am aware of the time. But now we're seeing where our terms are hitting. And there is I'm pretty certain that, yeah, I'm positive there's a YouTube video that NCBI has created about how to set up highlighting in your MyNCBI account, and also how to get a MyNCBI account. So if people have questions about that, they can consult that. But basically, I'm now putting my display to show the abstract. I have my highlighting turned on, and I can see we're hitting ulcerative colitis, we're hitting organoids. These terms are hitting, and the papers are making some sense. Now, we don't have time right now to really dive into these to see how relevant they are, but just eyeballing them, they seem to be relevant. And I'm seeing my terms hitting in parts of the record that are important for me, like the abstract and the title. I will also say, for example, that this paper has no mesh terms assigned to it. Aha! So here is proof that not every article has a mesh term assigned to it. And then here is, we go down one more, or a few more, actually. Sorry. Here, we can expand this section of the PubMed record and see where the mesh terms are listed and where the our terms are hitting on the mesh terms. So this is what I was referring to before in the mock-up slides of where the mesh term section of the record is. Okay, quickly, I'm going to do an experiment. And then I'm gonna open it up for questions. But my experiment is, suppose I just did a quick and dirty search, Rob. Maybe that's good enough. maybe you have just gone through this entire 2-hour presentation for nothing because PubMed's going to give me what I want. I've used PubMed all of my life. It works fine. I'm going to do what might be considered sort of a naive search, an unstructured search. And I don't mean naive in any pejorative way. I mean, this is how people search, right? This is what happens. But it's going to definitely be unstructured. No quotation marks, no Boolean operators, just a bunch of terms. And PubMed can do stuff with this, actually. And you know this, because you search PubMed probably like this, or have at least, for years. And it gives you useful results. So let's add this to the history. And we see we get a fair number, and we get hundreds. But we don't get 582. We get 469. So let's interrogate this a little bit and do an experiment where we say, are these 582, are there unique relevant papers in this 582 result set that I would not have found with this sort of unstructured naive search? That's the question. My hypothesis is yes, there is relevancy in these 582 that are not in the 469. But let's test that hypothesis. I'm going to say number 3, and then I'm going to use the Boolean not and say number 4. So this is an example. You could use actions, and actions has a and with add with not. But just I want to demonstrate another way you can do this, where you can use this abbreviated sort of placeholder or whatever, or representation. You can just say number 3, not number 4. And that will tell us what was not found by search number 4. And you can do this same thing in terms of terms. You add us some terms, you see what's the impact of that. It gave me 50 more results, but are any of those relevant? You can use the same not technique to answer that question. So we get 139. And the question is, are these 139 actually relevant? Maybe it's a bunch of noise. So we look at the 139, we look at the abstracts, and we see if there's evidence to support my hypothesis. And again, this isn't, we don't have time to really interrogate this fully, but I am seeing papers that look pretty on target to me. They're about inflammatory disease, about organoids used in the methods sections. So I'm going to just say, yeah, these techniques are useful and will, add relevance. Okay, that leaves all of 2 minutes for questions. My apologies for that. But, we've been answering questions all along, so I don't feel horrible about it. But
do any of the tools you have mentioned capture any gray literature?
Awesome question. You all are just like on fire with these questions. And I'm going to go back. This is a horrible way to go back, so I'm not going to do that. I'm going to go back up to here. So the short answer to your question is no. PubMed is not. There might be a little bit of gray literature. There might be some governmental reports in there. But I'm going to say probably not. PubMed is really not known as a great gray literature source. Yeah. But I will say, start this from here, that there are a number of sites here that are good for gray literature. So all of these in this category of curated collections also curate gray literature. So these 3 are good sources. Smafira is basically a machine learning tool for searching PubMed. So the answer would be the same for PubMed, that Smafira is really not a great tool for accessing the gray literature. Of the bibliographic databases, I would say Web of Science and Scopus are good for gray literature. There is some patent information at least in Web of Science. There's also conference proceedings and conference abstracts in Web of Science and Scopus. And those are gray literature sources. Those don't typically, they may or may not actually be parts of publications. And so those are also good sources for gray literature. And I know I said I had a hard stop at noon, but I'm good. We have just a few more questions, right? Can you say that one more time?
Are there any databases with mesh terms that contain unpublished information? Or are there any plans, at least that you know of, to develop something like this?
So published, I guess, is somewhat of a loaded word. I mean, gray literature is capturing things that are published per se outside of the sort of commercial publisher realm. So that would be like white papers or governmental reports, things like that. I will speak a little bit to the preprint world. So preprints are also could be considered published in the sense that they are part of preprint repositories, but could also be considered unpublished in the sense that they're not, part of, a journal. And PubMed does include preprints now. And I don't know if those are indexed. In other words, I don't know if preprints have mesh terms assigned to them, but they may. I'm not, actually, I suspect they are not mesh indexed. And just for a clarifying point, mesh terms are really used in PubMed as mesh terms are used in some other databases like the Cochrane database and also some sort of, how do I want to say, allied health, like CINAHL uses some mesh terms and PsycInfo uses mesh terms. But I would say that sort of PubMed is the kind of core source for the use of mesh in database searching, if that makes sense. So yeah, hopefully I answered that.
Yeah, I have a few more. Someone follow up to the gray literature question is, how can one's own gray literature be added to these search engines?
Oh, well, that's a good question. Not one I'd really thought about. I'm thinking, for example, of the inventory of 3R's knowledge sources. In particular, I know that they will link to organizational pages and also potentially kind of white papers or research findings from those organizations. And I assume that there is a scoping exercise, just like PubMed does a scoping exercise about what it is going to include, that the inventory of 3R's knowledge sources probably has a similar thing. I am not affiliated with this tool and I am speaking off the cuff, so I'm not 100% sure, but you may want to reach out to, I think the larger message is you may want to reach out to these repositories and say, hey, I have something that may be of value to this repository that I want to share with the broader research community. What are your, what are your rules around inclusion of materials? And, do you think my, white paper or whatever, it meets those inclusion criteria, and have a conversation with them about adding to those repositories. That would be my advice there. Yeah.
There was somebody said in the Q&A that there's partly free search access for gray literature on policycommons.net. I don't
Oh
know.
yeah, Policy Commons. Yeah, we just acquired that tool here at our library. So yeah, Policy Commons I think is a great one for the kind of regulatory testing kind of space. Yeah. And maybe there's time for one more. I do need to hop off.
we have a few more, but maybe this one, because I think this comes up a lot. Why relevant publications are not found in the quick and dirty search, although the term organoid is highlighted in yellow in the abstracts in the list of the missing publications.
Yeah, so just to clarify, the experiment I did here was that was comparing two strategies. And so what you're seeing here that are relevant are papers that were only found by the advanced search. Basically, were only found by the search that included mesh terms and field tags and Boolean operators and all of that stuff. So these were not at all missed by the exhaustive search. On the contrary, they were missed by the more naive or basic search. So that, I think, answers that part of the question, but I've actually now forgotten the second part of the question. If you could read that one more time.
Apologies.
Yeah, no problem.
There is actually not really a second part to the question. The person was just not understanding why the abstracts are then missing in the list of publications.
Right. So I think I answered that.
Yeah, you did answer it already. Yeah.
Okay, good.
Yeah.
Awesome.
Yeah.